Okay guys, so I thought I'd just do a really quick video on chi-squared. The reason I'm doing a video on this is because a lot of people have been saying to me, oh sir, we really don't understand what it is. Just looking from the book, it's very hard to follow what's happening, but to be honest with you, it's a fairly straightforward statistical analysis tool. So to begin with, it is pronounced chi-squared, not chi-squared, as a lot of people have been saying to me. This is something that I often got confused with as well, to be honest with you, until I heard somebody say to me, oh, it's not chi squared, it's chi squared. But before we start, let's get a couple of definitions out of the way, as we always do. A hypothesis, first of all, is something, it's sort of like a prediction, where we say when something happens to one variable, this will happen to another. So we could say that a definition of our hypothesis is a prediction of what will happen. So a prediction of what will happen depending on variables. Now this is like what you did at GCSE. We could say that when something happens to the independent variable, that will have an effect on the dependent variable. So if there's two variables at play and they're interlinked, if they're statistically viable, we say that that's our hypothesis. What's important in biology, and particularly when we're using statistics, is something called a null hypothesis. Now, if something's null, that basically means it's fake or that it does not exist. So a null hypothesis is always, no matter what it is, that any statistical significance is down to chance. So any statistical significance is down to chance. Now, what this means is if we're looking at a given variable, the variables are not really having an effect on each other. Any relationship that we see between the two is because of chance, because of things that have happened other than the variables affecting each other. Now, null hypotheses can be used both in looking at biodiversity and genetics. So we could say our null hypothesis when we're looking at genetics is that any genetic basis is not affecting the phenotype. What's actually happening is anything that we see is due to chance. However, there are certain states where we say that the hypothesis is true. Any variables are affecting each other and the phenotypes that we are seeing are not due to chance. So how does this work in genetics? This is where you need to understand how to use a chi-squared test in order to look at any two variables. So if we start with, if I clear this, don't save, if we start, for example, by looking at short and tall plants, okay, and if we say that the genotype is in blue and we say that the phenotype is in red. Okay, so genotype in blue, phenotype in red. Just to remind you, the genotype is the genetic makeup of an organism, whereas the phenotype is the visible characteristics that we can see. So I want you to imagine that we've got two heterozygous plants, and they're heterozygous for the tall and short gene. We're going to say that the short plants are recessive. Okay, so short plants are homozygous recessive. Therefore, Any plants with the dominant tall plant gene are tall. So if we do this as a Punnett square and we cross two heterozygous plants, so one plant is going to have the tall gene and the short gene. Same for the other one, tall gene and short gene. Okay. If we then do a genetic cross of these to develop our genotype, remember this is a monohybrid cross, we've got two dominant genes here, so this plant would be homozygous dominant. This one would be heterozygous, so it would have one dominant, one recessive. This one would also be heterozygous, so one dominant, one recessive. However, this one here would be homozygous recessive and therefore would be short. So if we look here, the phenotype for this one would be short, whereas all the others would be tall because they all have this dominant gene, okay, which sort of counteracts any effects of the recessive one they have. So from this we can see, or we can predict, we would expect that 75% of any population would be tall because we've got 1, 2, 3 that are tall out of 4, that is 75%. So 75% would be tall. Whereas 25%, the remaining 25%, would be short. So we would expect that any population of this plant would have 25% short. So 25% of the plants would be short. So for example, this would be our expected if we were looking at a population of 1,000 plants. So if we looked at a population of 1,000 plants, we would simply expect that 750 were tall, whereas 250 would be short. We can think of this as our hypothesis. We can say that our hypothesis is that if the short gene for a plant is caused by a homozygous recessive alleles, we could say that in any population, 25% of them would be short, whereas 75% of them would be tall. This would be deemed our hypothesis. Okay? Whereas if this isn't true, we could say that in any population that all the differences seen is due to chance. That would be our null hypothesis. So where we do a chi-squared test is where we want to either accept our hypothesis that is due to this monohybrid cross and the fact that two heterozygous parents are giving this percentage of population, or do we accept our null hypothesis and say that any differences between them all is due to chance? Chance mutations, chance changes in the environment. So if we look at if we change this and we start a new one, we don't want to save that. We want to don't save. We could start by looking at the chi-squared. So a chi-squared equation is as follows. We've got chi-squared, which is usually shown like this, is equal to the sum of the observed minus the expected squared divided by the expected, like so. Okay, so this is equal to chi-squared this is sum of, so essentially you're adding things together. This is the observed. This is the expected. So we've just worked out our expected for tall and short plants. We said it was a 75-25% split. So that's our expected. And it's just a case of being given the values and adding it to this equation, inserting it into this equation. So we've just worked out, if I put my Punnett square back up, if I draw it again very quickly, we've just worked out, or we've just said, that for plants, we could have a heterozygous and another heterozygous, and if we cross them, we would expect that 25% would be short if the height, the shortness of a plant is due to it being homozygous recessive. Okay? So we've worked that out here. We've said that 75% tall, we would expect, and 25% short. Okay? So if we were to get some real data, we could put it into a table as so. The way I always do a chi-squared test is by putting it into a very similar table. So we could, here along our rows, we have our phenotypes. So the two phenotypes we've got are tall plants and short plants. We could then put our observed, which is O. We could then have our expected. So expected. We could then have our observed minus our expected. Then our observed minus our expected squared. Put that in brackets, because remember, BIDMAS, we always do the brackets first. And then we could finally have the whole equation. We could have O minus E squared divided by E. And I always arrange my chi-squareds into a table like this, purely because if you try to cut corners, either you can get confused or the calculator can get confused. So I want you to imagine that we've got an observed amount of plants, tall and short plants. 590 were tall, whereas 174 were short. Now, if we add those together, quickly grab my calculator because my maths is not absolutely fantastic. 590 plus 174 equals 746. So that's a total of 746. Now, we said earlier, by doing our monohybrid cross, that we would expect that 75% would be tall and 25% would be short. However, 75% of 746 equals 560. Okay? So our expected would be 560, whereas our observed would be 590. So for our short plants, our expected would have been 186. Now you can see that although they're not far off, they're not exact. These numbers are not exact. Our expected is not the same as our observed. So we're not sure here whether this is due to chance or due to the fact that there are variables at play between the genes. So once you've got your observed, and once you've done a Punnett square or a genetic diagram to work out your expected, you can simply fill in these. So 590 minus 560 is 30. And then 174 minus 186 is minus 12. Then what you need to do is you need to square these numbers. So 30 squared, if I just punch that into my calculator, equals 900. Whereas minus 12 squared equals 144. Okay? So then in this column, we would do 900 divided by 560. So 900 divided by 560 equals 1.6. Obviously that's rounded. Whereas 144 divided by 186 equals 0.7 or 0.77. So we'll round it 0.8. Okay? Now remember that chi-squared asks you for the sum of. So the sum of this is 1.6 plus 0.8, which gives you a sum of 2.4. So our chi-squared value is this number plus this number, which gives us a value of 2.4. Chi-squared for us is 2.4. Now, that's all well and good. We've worked out our chi-squared value, but what does this number mean? We need to add some sort of value to this number. Otherwise, it's just 2.4 and it doesn't actually mean anything. Okay? Now, this is where we need to take into account something called critical values. So once you've worked out your chi-squared value, you need to compare it to a table of critical value. Okay? So 2.4 is our chi-squared. So when I make this new, I'm going to write that at the top dead quick. So our chi-squared is 2.4. We've just worked that out by using our expected and our observed values. So, as I just said, you need to compare this to something called a table of critical value. Now, a table of critical value simply looks like this. You'll have the first column that says degrees of freedom, and you'll have a second column that has your critical values on. Critical values. Okay? Now, in this critical value column, you'll see that there's various numbers. Those numbers will be 0.5, 0.2, 0.1, 0.05. And sometimes you'll have more than that. However, it doesn't matter because the only column we are ever interested in is the 0.051. What this tells us is that any value here is 5% due to chance. We're looking at the 5%. If we look at the 10% or the 2% or the 50%, in biology, these are not of statistical significance. They may be in other uses in statistics, but in biology, the only critical value column we are bothered about is this 0.051, this 5% chance. Okay? So essentially what this means is that there's a 5% probability chance that our results are due to chance. Okay? Now in any exam, you'll always be given this table. You'll always be given a table of critical value. You don't need to know this off by heart. And the 0.05 column will have the following numbers. It'll have 3.84, 5.99, 7.82, 9.49, and it'll keep going. Okay? However, we don't need to write all those out because you'll see in a minute what we actually want them for. So before I go any further, let me remind you, in terms of biology, in terms of your A-level examinations, you can always ignore these columns. You can always ignore the 50%, 20% and 10%. The only column we are ever bothered about, because in biology is the only column that has statistical significance, is the 5% chance, the 0.05 column. That's because this means that there's only a 5% probability that our results are due to chance. Now, the other column that you need to know about is your degrees of freedom column, and this will simply be a 1, 2, 3, 4. Okay, and these are in line with these numbers here. Now, your degrees of freedom is calculated by the number of phenotypes possible, so the number of phenotypes possible, help if I could spell possible, minus 1. So, for example, in our plant example that we did, where we worked out our 2.4, there were two phenotypes. There was either tall plants or short plants. So we would simply do 2 minus 1, which gives us 1, and that will give us our degrees of freedom. So our degrees of freedom is calculated by the number of phenotypes possible minus 1. So we had tall or short plants, 2, minus 1 to give us 1. So the column we're concerned with is this one here. Okay? We go across from the 1, and remember we're only using the 5% chance due to probability. If this was a different test, say for example if you were looking at the possibility of numbers turning up off a dice has 6 faces, therefore the possibilities you could have are 6. 6 minus 1 is 5, therefore you would use the 5 degrees of freedom. However, we're only looking at 2, so you use the 1 column. Okay? So, if we use the degrees of freedom column 1 and go across to our 5% chance, this gives us a number of 3.84. So the number we're looking at is this one, 3.84. Now this is a number that we can use to compare our original chi-squared of 2.4. Okay? And as you can see, our number, 2.4, is lower than the 3.84 column. Okay? Therefore, what this is saying is if it is lower, if our number is lower than this critical value, we must accept our null hypothesis. We accept our null hypothesis. And therefore, we can say that our observed results are due to chance. There's no statistical difference between what we expect and what we see. Okay? So, if it's higher, so if it was higher than this, we would reject our null hypothesis, and therefore the variables would affect each other. So if it's higher, we reject the null hypothesis. Okay? But because our number is lower, we accept our null hypothesis, and therefore there's no statistical difference between what we expect and what we see. Okay? So that's essentially your chi-squared test. This is how we carry it out. I hope this has been of some use to you. If you need any help, come and see me. Okay? Thank you.